Welcome to this deep dive into the most surprising model-changing longevity research published between February 13th and February 20th, 2025.
Absolutely.
We've curated this specifically for longevity biotech researchers and experts like you,
Yeah,
pulling out the most impactful papers from credible institutions.
right.
So get ready for some ha moments as we unpack findings that could reshape your understanding of aging and its potential interventions.
It's a really interesting match this week.
Yeah.
With some findings that could be game changers in the field.
I'm with you there. For example, this paper published just today in Nature Medicine really challenges a core assumption about aging.
Right, integrating the environmental and genetic architectures of aging and mortality from the Nuffield Department of Population Health at the University of Oxford.
Okay?
Instead of asking nature versus nurture, this paper quantifies each side of the equation in a really compelling way.
And the findings are striking, using data from the UK Biobank.
Yes.
The authors suggest that the environmental component, what they call the exposome, has a surprisingly large impact on our mortality.
It's a massive difference. The exposome accounted for an additional 17 percentage points of mortality variation, while genetics contributed less than 2.
Wow.
While we can't ignore the impact of certain genetic conditions on lifespan, this research suggests that focusing on environmental factors could be a more effective path to extending lifespan and preventing age-related disease.
Which has huge implications for everything from public health initiatives and lifestyle choices to how we design our cities.
Yeah.
What are your thoughts on the robustness of their exposome definition and analysis?
They've taken a very comprehensive approach, incorporating a wide range of environmental factors, including socioeconomic status, lifestyle habits, and even exposure to pollutants. It's a much more nuanced view of the exposome than we've seen in previous research.
It definitely makes you think about how we approach longevity interventions. Maybe instead of searching for that single magic pill, we should be focusing on creating environments that support healthy aging.
That's a great point, and it leads perfectly into a couple of papers, both published today in Nature Aging, that highlight a specific intervention that's generating a lot of buzz in the longevity community.
Okay.
FGF21.
Fibroblast growth factor 21, it seems to be popping up everywhere in longevity research lately. What's driving all the excitement?
One of the biggest challenges in aging research is finding ways to combat age-related immune decline.
Right.
These two FGF21 papers offer a potential solution by targeting something that's long been considered irreversible.
Yeah.
Thymic involution.
Which is a big deal because the thymus is essentially where T cells crucial for a robust immune system mature. As the thymus shrinks with age, our immune response weakens, leaving us vulnerable to infections and diseases.
Exactly, and what's remarkable about these papers is that they show in mice that manipulating FGF21 signaling can actually regenerate the thymus, leading to improved immune function in older animals.
Wow.
The first paper from UT Health San Antonio, Paracrine FGF21 dynamically modulates mTOR signaling to regulate thymus function across the lifespan, delves into the molecular mechanisms behind this regeneration.
And the second paper, Enhanced Paracrine Action of FGF21 in Stromal Cells Delays Thymic Aging from Yale, confirms these findings, demonstrating that elevating FGF21 in specific thymus cells can protect against age-related decline and even extend health span.
Right.
It seems like we're seeing convergent evidence here from multiple respected institutions.
And what's really intriguing is how this research on FGF21 ties into the bigger picture of longevity. We already knew FGF21 played a role in metabolism and longevity.
Right.
Now, these studies add another layer to the puzzle, suggesting that it could be a key player in maintaining a healthy immune system as we age. That kind of multifaceted impact is exactly what we look for in potential longevity interventions.
It makes you wonder what other surprises FGF21 might hold. What are your thoughts on the potential for translating these findings into therapies for humans?
It's still early days, of course. These are preclinical studies in mice, but the results are certainly promising and warrant further investigation.
Okay.
Given the challenges of regenerating organs, this is definitely a finding that could change how we think about treating age-related immune decline.
Absolutely. Now, another paper published today, this one in Geroscience, caught my eye because it focuses on a clinical trial design, which is exciting to see.
Test of rapamycin in aging dogs, TRIAD, is a groundbreaking trial that could have major implications for human longevity research.
Okay.
It's testing the effects of rapamycin, a drug already known to extend lifespan in mice, but this time it's being studied in dogs.
And using dogs as a model for aging research is fascinating because they experience many of the same age-related diseases as humans, like cancer, heart disease, and cognitive decline.
Right.
Plus, their shorter lifespans allow us to observe the effects of interventions more quickly.
Right. And this trial is particularly important because it's the first time rapamycin is being tested in a larger, more complex mammal that shares our environment.
If it proves successful in dogs, it could pave the way for human trials and potentially revolutionize how we approach aging.
It's incredible to think that man's best friend could be a key to unlocking longer, healthier lives for humans. I'm really curious to see how this trial unfolds.
Me too. Speaking of how different parts of the body age, there's a paper characterizing brain cardiovascular aging using multi-organ imaging and machine learning published today that uses machine learning to analyze imaging data from the UK Biobank. And what they found is a strong link between the aging of the cardiovascular system and specific brain networks.
Which really emphasizes that aging isn't just about one organ deteriorating in isolation, it's a whole body phenomenon. And this paper highlights the importance of taking a more holistic view of aging research, investigating how different systems interact and influence each other over time.
Absolutely. It's not enough to just focus on one organ or system. We need to understand how the aging process unfolds across the entire body and develop interventions that address these interconnected changes. And this paper also showcases the incredible potential of machine learning and AI in longevity research.
Right.
It's allowing us to analyze complex data sets and uncover hidden connections that we might have missed using traditional methods.
Right. I think we're just scratching the surface of what these technologies can do for longevity research.
Well, let's move on to a topic that's been generating a lot of excitement in the field, senolytics. We have a whole set of papers this week exploring new ways to target senescent cells and their harmful effects.
Senolytics are definitely a hot topic right now, and for good reason. They offer a targeted approach to addressing one of the hallmarks of aging, cellular senescence.
Right. Senescent cells, or zombie cells as they're sometimes called, have stopped dividing but refuse to die off. They accumulate as we age and release harmful substances that contribute to inflammation and tissue damage, driving various age-related diseases.
And the exciting thing about senolytics is that they can selectively eliminate these harmful cells, potentially slowing down or even reversing the aging process. And one of the papers published this week, AI-driven robotics laboratory identifies pharmacological TNIK inhibition as a potent xenomorphic agent, demonstrates just how powerful this approach can be.
This paper published in Aging and Disease from researchers at In Silico Medicine is fascinating because they used an AI-powered robotic lab to identify a completely new xenomorphic drug.
It's a great example of how AI and automation are transforming drug discovery. This new drug called INS0188CO55 targets a protein called TNIK and has shown very promising results in preclinical studies reducing cellular senescence and inflammation.
That's really impressive. What are your thoughts on the potential of this particular drug, especially given the AI-driven discovery process?
It's definitely a novel approach, and the preclinical data looks promising. Of course, we need to see how it performs in human trials, but it highlights the potential of AI to accelerate the discovery of new and effective senolytics.
Another exciting paper. Targeting CAP2 in a novel senolytic strategy prevents glioblastoma recurrence after radiotherapy, published in EMBO Molecular Medicine from the University of Texas Health, explores using senolytics to combat cancer.
This paper highlights the connection between aging and cancer and how targeting senescent cells could potentially address both. They found that by targeting a protein called CIAP2, they could eliminate senescent glioblastoma cells and prevent tumor recurrence in mice.
It's another example of how senolytics could have a double whammy effect, combating both aging and age-related diseases.
And finally, we have a preprint from the Center for Oral Immunology and Regenerative Medicine at Queen Mary University of London titled Membrane Transporter Progressive Ankylosis Protein HAMMOG partially mediates senescence-derived extracellular citrate and is regulated by DNA damage, inflammation, and aging. While it's still undergoing peer review, it offers some intriguing insights into how senescent cells communicate during aging.
It dives deep into the molecular mechanisms, focusing on a protein called ANKH. What's fascinating is that ANKH seems to be involved in the buildup of citrate around senescent cells, which appears to be linked to various age-related conditions.
It's like they're sending out signals that contribute to the dysfunction we see in aging tissues, and by understanding the role of ANKH in this process, we could potentially develop new senolytic therapies that interrupt this communication system and prevent some of the harmful effects of senescent cells.
It's amazing how much we're learning about senescent cells and their role in aging. I have a feeling we'll be hearing a lot more about senolytics in the years to come, but for now, let's move on to a few other papers that caught my attention exploring different aspects of aging. One that I found particularly interesting was chronic adrenergic agonist treatment improves neurovascular coupling responses, attenuates blood-brain barrier leakage and neuroinflammation, and enhances cognition in aged mice.
published in Aging from the University of Oklahoma Health Sciences Center. This study offers a potential new approach to addressing cognitive decline. They found that stimulating a specific type of receptor, the B3 adrenergic receptor, could improve brain blood flow and reduce inflammation in aging mice.
It's like giving the brain a much-needed boost. And what's particularly exciting is that this approach could be less invasive and potentially safer than some other cognitive decline therapies being investigated.
Yes, and the fact that they saw improvements in neurovascular coupling, blood-brain barrier integrity, and neuroinflammation suggests that this approach could have broader benefits for brain health beyond just cognitive function.
Now let's shift gears and talk about a preprint from Harvard T.H. Chan School of Public Health titled Peroxisomal Dysfunction Drives Loss of Dynamic Nutrient Responses to Initiate Metabolic Inflexibility. During aging, this one caught my eye because it takes a deep dive into how our cells adapt to changes in nutrient availability, a process that becomes increasingly important as we age.
This research highlights the crucial role of these tiny structures within our cells called peroxisomes. They've discovered that peroxisomes are far more important in this adaptation process than previously thought. And perhaps even more exciting, they found that dietary restrictions seems to have a rejuvenating effect on peroxisome function.
Which adds to the growing body of evidence suggesting that dietary interventions can significantly impact how we age. What are your thoughts on the potential of targeting peroxisomes for longevity interventions?
It's a relatively new area of research, but the findings from this study are compelling. If we can develop strategies to maintain or even enhance peroxisome function as we age, it could have a profound impact on metabolic health and potentially slow down the aging process.
Now, another preprint, this one from the Indian Institute of Science Education and Research, Mohali, caught my attention because it explores the surprising connection between iron and aging. Iron-to-peat diet enhances Canohabditis elegans lifespan via oxidative stress response pathways, suggests that reducing iron intake can actually extend lifespan in these tiny worms.
It's a thought-provoking study because it challenges our assumptions about the role of iron in aging. While iron is essential for many biological processes, it can also contribute to oxidative stress. And this research suggests that reducing iron levels, at least in this specific model organism, can trigger a protective stress response that ultimately promotes longevity.
Of course, we need to be cautious about extrapolating these findings to humans, but it does make you wonder whether tweaking our iron intake could have any impact on lifespan and health span.
It's definitely an area that warrants further investigation, and it highlights the importance of understanding the delicate balance of nutrients and how they influence the aging process.
Okay, one more preprint before we move on to the final set of papers. This one comes from CRS University Lyon 1 and focuses on the physical forces at play during aging. It's titled, Putative interfollicular stem cells of skin epidermis possess a specific mechanical signature that evolves during aging.
This is a great reminder that aging isn't just about what's happening inside our cells at the molecular level, but also about the physical forces that influence tissue structure and function. This paper shows that stem cells in our skin have a unique stiffness that changes as we age.
And this changing stiffness could affect how well our skin renews itself, contributing to wrinkles and other age-related skin changes.
Exactly. It's a fascinating area of research, and it underscores the importance of understanding how biophysical changes contribute to the aging process.
Now, before we wrap up this section on preprints, there's one more that I wanted to highlight. The nuclear pore complex connects energy sensing to transcriptional plasticity in longevity, published on bioRxiv from researchers at Massachusetts General Hospital, dives into the intricate world of cellular communication.
They found that a specific protein complex, the nuclear pore complex, plays a key role in linking energy sensing to longevity in the model organism C. elegans.
Which is interesting because we usually think of the nuclear pore complex as a gateway for molecules to move in and out of the nucleus, but this research suggests it also plays a role in regulating lifespan.
It highlights the interconnectedness of cellular processes and how even seemingly mundane functions can have a profound impact on longevity.
It's amazing how much we're still learning about the complexities of aging.
Indeed, each discovery seems to add another layer to this intricate puzzle. Now, are you ready to dive into the final set of papers for this week?
Absolutely. Let's hear what other surprises the world of longevity research has in store for us.
Okay. This next paper, published in Telomerase, dives into the genetics of aging and its impact on both longevity and cancer risk. It's titled, Genetic regulation of TERT splicing affects cancer risk by altering cellular longevity and replicative potential.
TERT, the gene responsible for producing telomerase, has long been a focus of aging research. Telomerase, of course, plays a key role in maintaining the length of telomerase.
Right.
Those protective caps on the ends of our chromosomes that shorten as we age.
And what this research uncovered is that certain genetic variations in the TR gene can affect how telomerase is produced, which in turn influences telomere length and cellular longevity. Essentially, some individuals might be genetically predisposed to have longer telomeres, potentially leading to a longer lifespan.
But as we know, it's never that simple.
Right. The researchers also found that these same genetic variations can impact cancer risk. Some variations might offer protection against certain cancers, while others could increase susceptibility. It's a complex interplay between longevity and cancer risk, highlighting the need for a more nuanced understanding of how genetics influences the aging process.
It's almost like a genetic balancing act where certain variations promote longevity but come with an increased risk of other age-related diseases. This research really underscores the complexity we're up against in unraveling the aging process.
It definitely adds another layer to the puzzle. And speaking of complex systems, let's move on to a paper published in Aging Cell titled The Impact of Toll-like Receptor 5 on Liver Function in Age-Related Metabolic Disorders.
This one caught my eye because it focuses on the intersection of the immune system and aging. Specifically, the researchers looked at toll-like receptor 5, or TLR5,
Right.
which plays a crucial role in our immune response to pathogens.
And what they found in mice is that TLR5 deficiency leads to a cascade of metabolic abnormalities as they age, particularly in the liver. These mice showed an increased tendency for liver fibrosis and fat accumulation.
A weakened immune system, in this case caused by TLR5 deficiency, can contribute to metabolic dysfunction and age-related diseases like liver disease. It's another example of how interconnected the aging process is and how a decline in one system can have ripple effects throughout the body.
Absolutely. This research adds to the growing body of evidence suggesting that maintaining a healthy immune system is crucial for longevity and health span. Now, before we move on, there are a couple of interesting papers that deserve a quick mention. One is from eLife titled, Flavin-containing monoxygenices regulate longevity and health span through xenobiotic detoxification and riboflavin metabolism.
This one piqued my interest because it explores the role of flavin-containing monoxygenases, or FMOs, in longevity and health span. These enzymes are involved in detoxification and metabolism, and the researchers found that FMOs are upregulated in multiple longevity interventions, suggesting a protective role against aging.
It's intriguing research that opens up new avenues for exploration. Could boosting FMO activity be another strategy for extending lifespan? It's certainly a question worth investigating.
Absolutely. And the other paper I wanted to mention briefly is from Nucleus Pulposus titled circ-FNDC1 interacts with CDK9 to promote mitophagy in nucleus pulposus cells under oxidative stress and ameliorates intervertebral disc degeneration.
This one dives into the molecular mechanisms behind intervertebral disc degeneration, a common age-related condition that causes back pain. They identified a circular RNA molecule called circ-FMDC1 that seems to protect against oxidative stress and degeneration in the discs.
Which is exciting because it could potentially lead to new therapies for this debilitating condition. It also highlights the growing importance of understanding the role of non-coding RNAs in aging.
Right. For a long time, we focused primarily on protein-coding genes, but now we're realizing that these non-coding RNAs like circ1 and NDC1 play a critical role in regulating gene expression and cellular processes, and they may hold the key to unlocking new longevity interventions.
It's another reminder that we're just beginning to unravel the complexities of aging. There's so much more to discover.
Now let's wrap up this week's deep dive with a paper from the Journal of Cerebral Blood Flow and Metabolism, Dynamic Diffusion-Weighted Imaging of Intracranial Cardiac Impulse Propagation along arteries to arterioles in the aging brain, caught my eye because it uses a novel imaging technique to study how the heart's impulse travels through the brain's blood vessels.
And they found that this impulse propagation changes with age, which could have significant implications for understanding vascular health and cognitive function as we age.
It's a fascinating study that provides valuable insights into the intricate relationship between the cardiovascular system and the brain during aging.
But it looks like we've covered quite a diverse array of papers this week, from the genetics of aging to the role of the exposome, from senolytics to FGF21, from the complexities of the immune system to the biophysical changes in our skin. It's remarkable how much progress is being made in the field of longevity research.
It's truly an exciting time to be following this field. The pace of discovery is accelerating, and it feels like we're on the cusp of a longevity revolution.
I completely agree. Now, before we wrap up, I'd like to hear your thoughts on what all of this research means for the future of aging. Are we really on the verge of extending human lifespans significantly?
I think we are moving beyond simply accepting aging as an inevitable decline and towards a future where we can actively shape our health span and extend the number of healthy, vibrant years we live. The research we've discussed today highlights the incredible plasticity of the human body and its remarkable capacity for repair and rejuvenation.
It's almost like we're rewriting the rules of aging, challenging the very notion of what it means to grow old.
I'd say we're moving from passive passengers to active participants in the aging process. We're taking control of our health, empowering ourselves to live longer, healthier lives. And that's what makes this research so compelling. It's not just about adding years to life, but adding life to years.
Beautifully said. Now, before we sign off, there's one more paper we should mention, a roadmap for alleviating the manifestations of aging in the cardiovascular system, published in Nature Reviews Cardiology.
It's a timely paper that provides a comprehensive overview of the challenges and opportunities in cardiovascular aging research. And it stresses the importance of a multi-pronged approach addressing everything from lifestyle factors to cellular mechanisms to emerging therapies.
What are your thoughts on the key takeaways on this roadmap, especially for researchers and clinicians working on the front lines of cardiovascular health?
I think the emphasis on personalized interventions is crucial. Aging is a highly individual process influenced by a complex interplay of genes, environment, and lifestyle choices. What works for one person might not work for another. So moving forward, we need to focus on developing tailored approaches to cardiovascular health that take into account an individual's unique needs and circumstances.
It's a call for a more nuanced, personalized approach to medicine, moving away from one-size-fits-all solutions and towards tailored interventions that address the root causes of aging and disease.
Exactly, and that's where I see the most potential for making a real impact on human health span. It's about understanding the individual, not just the disease.
It feels like we're on the verge of a real paradigm shift in how we approach healthcare, moving from a reactive model to a proactive one, focusing on prevention and early intervention rather than just treating diseases after they've already developed.
I agree, and this shift is especially critical in the context of aging, where we're dealing with complex, interconnected processes that require a more holistic approach.
It's not just about adding years to life. It's about adding life to years, ensuring that those extra years are healthy and fulfilling. And that's where I think this research on aging is having a real impact. It's changing how we think about the possibilities of extending human health span.
Absolutely. We're moving beyond simply accepting aging as an inevitable decline and embracing the idea that we can actively shape our own aging trajectory.
And what's really exciting is that this research isn't just happening in labs and academic journals. It's translating into real-world interventions and products that can make a tangible difference in people's lives.
We're starting to see senolytics enter clinical trials, personalized nutrition plans based on individual genetic profiles, and even AI-powered fitness apps that optimize exercise routines for healthy aging. The convergence of science, technology, and a growing awareness of the importance of healthy aging is creating a perfect storm for innovation in this space.
And it's not just about scientists and researchers. It's about empowering individuals to take control of their own health and longevity. From making informed lifestyle choices to participating in citizen science initiatives, everyone has a role to play in this longevity revolution.
I couldn't agree more. This is a movement that requires collective action, from individuals making healthy choices to policymakers creating environments that support healthy aging to researchers pushing the boundaries of what's scientifically possible.
It's a collective effort to rewrite the narrative of aging, shifting from a story of decline to a story of possibility and resilience.
Exactly. And that's what makes this field so inspiring. It's not just about science, it's about hope, empowerment, and the potential to transform the human experience of aging.
So as we wrap up this deep dive into the latest longevity research, what's the key takeaway message for our listeners? What should they be thinking about as we continue their work in this exciting field?
I think the main message is this: the future of longevity is in our hands. We have the knowledge, the tools, and the collective will to make a real difference in how we age. It's time to embrace this challenge and create a future where aging is no longer seen as a limitation, but as an opportunity for continued growth, discovery, and fulfillment.
Beautifully said. And on that inspiring note, we'll wrap up this episode of The Deep Dive. We hope you've enjoyed this exploration of the latest longevity research and are feeling inspired to continue exploring the fascinating world of aging. Until next time, stay curious, stay informed, and stay engaged in the quest for a longer, healthier life.